Welcome, everyone. Let me just start with some brief housekeeping items. Sorry, some behind the scenes logistics. So first, let me introduce myself. I always forget to do so. Hello, everyone. I'm Dr. Ilaria Bertoletti, and I am currently the program director for our PhD in Educational Neuroscience program and director of Myelin Lab. We are very excited to have our speaker with us today. Just for everyone to know, I want to acknowledge that we have interpreters here today as well as live captioning. If people want to share thoughts or chat, you can use the chat box, which is located in the chat feature at the bottom of the screen. That's just for people to share.
Can I interrupt? It seems like in the chat we're having some problems. It looks like they can only see the interpreter and they can't see Ilaria or Dr. Bertoletti.
Thank you for letting me know. I think I changed that now.
They say it's fixed. Yeah.
So sorry, everyone. Let me start over. Technology. Hello, everyone. I'm Dr. Ilaria Bertoletti. I'll get started with some housekeeping items. First, we have interpreters interpreting the talk today, as well as live captioning. And for people who want to share thoughts about the talk, they can use the chat feature. And that is just for social interaction among our audience members. If anybody, and if you have a technical problem, please make sure that you contact the host and panelists. Once the presentation is over, we will have a formal Q&A, and there is a specific Q&A feature at the bottom of the screen, so please type your questions there. I will also share a link to our webpage where you can see all of the historical talks from the Distinguished Lecture Series. And you will also be able to see today's presentation, which will be uploaded in a few weeks from now at that same link. I'd like to invite one of our students, Deanna Dunlop, to come up and give a formal introduction to our speaker today. Thank you, Deanna. Hello and welcome to the PhD in Educational Neuroscience Distinguished Lecture Series hosted by Gallaudet University. This lecture series aims to honor world-renowned scientists in the fields of psychology, education, cognitive sciences, and neuroscience. These different fields, and all of the interdisciplinary fields in between, contribute to the new and growing field of educational neuroscience. They increase our understanding of the human mind, as well as the neuromechanisms of learning. This year's Distinguished Lecture Series theme is exploring the foundations of human development and consciousness. I am honored to introduce our distinguished lecturer today, Dr. Allyson Mackey. Dr. Mackey is an associate professor in psychology at the University of Pennsylvania, and also serves on the Scientific Advisory Council of the National Center for the Developing Adolescent. Dr. Mackey's research has three primary directions. First, on how experiences during development can change the brain and how this changes the mind. Second, the effects of maturation on brain plasticity. And third, the function of environmental factors on windows of peak plasticity. She has received the Rising Star Award from the Association of Psychological Sciences and the Early Career Award from the International Mind, Brain, and Education Society. Today's presentation is titled Environmental Influences on Brain Plasticity. Now, please join me in welcoming Dr. Allyson Mackey. Dr. Mackey, the floor is yours.
Good afternoon, everyone, and thank you so much for having me. I want to take a moment to think about the many problems that our society faces. We can think about literal and figurative storms brewing. And we can think about how long these problems will take to solve. These problems will not be solved by us, but they might be solved by our children. So we can think about what are the skills that kids are going to need to solve these problems. They will need strong reasoning skills and creativity, curiosity, resilience. In my lab, we're interested in how the developing brain gives rise to these skills and the kinds of environments that shape the developing brain and the developing mind. We're particularly interested in the deltas or the changes. So how can we change the environment to better foster neurocognitive development? When I first started thinking about this question in graduate school, I thought about changing the classroom environment. In this work, we found that when kids play fun games to boost their reasoning skills, we found gains in IQ. We also found that when adults practiced for the law school admissions test, their brains changed in terms of their structure and their function. What we were observing in these studies was brain plasticity as a process of change. So we can think about how when cells fire together, they wire together. We see changes in synapse formation, changes in myelination, and changes in other glia. But what is so striking about studying interventions is the variety of responses. Some brains change a little, others change a lot. And so these results led me to wonder about plasticity as the potential to
Sorry.
change.
There's some technical issues with the vision. Can everybody see interpreter and presenter? Can someone type in the chat and let me know? Okay, it seems like, yes, sorry about that.
We're good?
Yes.
Okay. So we know that while myelination stabilizes connections and makes them faster, it also acts like glue in the sense that it prevents future change. We also know from animal models that inhibition increases over development and is a brake on plasticity, prevents plasticity. Still, critically, the brain sort of reserves the right to change throughout life. And we know, again, from animal models that dopamine can create juvenile-like plasticity even in adult brains. So taken together, these results suggest that maturation restricts plasticity through myelination and inhibition, and motivation can boost plasticity. So I'm curious about how brain maturation proceeds during childhood. I'm interested in how children's experiences impact the rate of maturation, how children's experience impact motivation, and what we might be able to do to enhance brain plasticity. We're studying humans, and so we are relying not on studying cells, but on MRI. And we scan young children, and young children tend to move in the scanner. And so we have a pretend scanner or mock scanner where they can practice being still and their movie turns off if they move their head too much. This usually works. Usually we can get good data from young kids, but I will say not always. So an ongoing challenge is figuring out how to design our sequences and analyses to handle young kids' motion. So even best case scenario, our images have big voxels or volume pixels. So you can see for scale, the cellular processes I was telling you about are quite small compared to the resolution of MRI. But what I like to argue is that while voxels are big, brains are bigger. So you can see one voxel here in white for scale. And this is important because there's more and more evidence suggesting that our brains are organized in these broad, widespread networks. Here I'm showing a gradient from areas in yellow that are responsible for processing sensory information, producing motor output, and areas in purple that are sort of disconnected from the real world, that process things that cannot be directly sensed, like the past, the future, the minds of other people. And so now for more than a decade, there have been ideas about how brain plasticity might change over development. Right? So we can put age on the x-axis and plasticity on the y-axis and make predictions that maybe sensory and motor plasticity might decrease faster than plasticity in association areas that are responsible for higher level cognition. And this tracks with experiences of having a harder time learning new languages or new sports after childhood. And our ability to learn new things like neuroscience all the way into adulthood. There have also been ideas about how these curves might look different in some individuals. So that experiences of, say, early life stress might accelerate these curves and limit windows of peak plasticity. While I think these theories are powerful, where I'd like to get to is actually putting numbers on these axes. How can we actually measure plasticity in developing humans? So now I'm going to talk with you about 3 approaches that my lab has taken. Starting actually with the newest work on measuring inhibition. What we know from animal models is that PV cells are particularly important for limiting plasticity. These cells control the output of neurons and make connections very near to the axon. These cells are also more prevalent in sensory areas than association areas. Another population, we'll call them SST neurons, make connections to more distal dendrites and are more prevalent in association areas of the brain. Both produce inhibition. So we've recently started thinking about a measure called the Hurst exponent to measure inhibition. And this idea comes out of a computational model initially. So in this model, we have sort of toy neurons, one that's excitatory, E, and one that's inhibitory, I. And in this model, you can change how active each of the neurons are. When there's higher inhibition, shown here in orange, there's higher simulated activity, neural activity in the system. This neural activity would lead to a change in blood oxygen level dependent or bold signal. So you can multiply the simulated neural activity with the hemodynamic response function to get these different signals. And then you can transform the signals to look at how much power there is at higher frequencies as compared to lower frequencies. And that can be measured with a slope called the Hurst exponent. So in blue, this is a case of lower inhibition. We have a lower Hurst exponent, more signal at higher frequencies. And in orange, we have a case with a higher Hurst exponent, less signal, less noise at these higher frequencies. But this is a phenomenon from a computational model. And we wanted to understand the empirical basis of this argument. And so we wondered whether there was a correlation between Hurst exponent and markers of inhibitory neurons across cortexes. So this is a spatial correlation. So ideally, we'd be looking at inhibitory cells, but these cells are classified based on the expression of protein. And what we actually have the best access to is RNA expression. So we can use a public data set called BrainSpan for each area of the brain to ask about the level of RNA for PV cells. The inhibitory cells we know are important for plasticity, as well as other types of inhibitory cells like the SST neurons. Let me show you what we're finding. Okay, so we're looking at PV expression in ex vivo brains. There are thankfully very few of these brains in childhood, but we can look at expression in different regions and ask how it correlates with Hurst exponent in our fMRI data. And what we find is that brain areas with higher PV expression also have higher Hurst exponent. This plot is in children. This plot is in adults. So again, we have PV expression from adult ex vivo brains and Hurst exponent from adult fMRI. You'll notice that the Hurst exponent is much higher in adults than in children, as we would expect. And we see the same correlation. Areas that have higher PV expression have higher Hurst exponent. This work was led by Monami Nishio, who's a second-year graduate student in my lab. Monami also wondered whether the same thing is true in animal models. And so she was able to find data on both fMRI from mice and rats, and also in mice, I'll show you the data from mice, data on the PV cell density. Here, brain regions in mice with higher PV cell density also have higher Hurst exponent when you look at mouse fMRI data. This is true for PV cells. It's not true for other types of inhibitory neurons. And so these data made Monami feel more sure that the Hurst exponent was a reasonable readout of inhibitory activity. And so she wondered then, how does the Hurst exponent change over development in humans? And this is what she found. So sort of regardless of where you're looking in the brain, whether it's sensory cortex or association cortex or somewhere in the middle, you see a similar kind of curve with an increase in the Hurst exponent until about age 8. The plateau is slightly earlier for both ends of this sensory association axis, a little later for mid-rank areas. This pattern of a plateau around age 8, we've now been able to replicate in a much larger data set. So we're feeling sort of more sure about this pattern. What's also very interesting is it looks like the pattern we're seeing in the Hurst exponent matches the ex vivo data. So if we go back to our mRNA markers of inhibitory neurons, we see both now for PV and for SST an increase in expression until about age 8 and a plateau after that, sort of regardless of where you're looking in the brain. So what is this inhibition for? One way to think about it is that inhibition can lead to functional specialization. Right? This inhibitory neuron might make this cell's pattern of activity more different from this cell's activity. Right? So sort of setting boundaries in cortex. So we think initially cortex sort of starts out pretty multi-purpose, and then our experiences and the increase in inhibition starts to set these kinds of boundaries. The way we've been thinking about functional specialization is looking at resting state fMRI. And the premise of this technique is that you can think about two regions, these sort of patches of cortex, that are in the same cortical network. It will say these areas and sort of the same network. What we'll find is that if you look at the pattern of fluctuations in blood oxygen for one area, they will be correlated with all other areas in the same network. If instead you look at two patches of cortex that are in different networks, what you might find is that their bold signals are anti-correlated. In adults, we know that areas that direct attention to the outside world, like areas here in green, are anti-correlated with the default mode network here in pink that mostly processes the internal world. So these seem to be fairly separate and anti-correlated in adults. In childhood, we see that these anti-correlations grow stronger. So the correlation between the default mode network and the dorsal attention external attention network gets stronger. Same is true for the anti-correlation between the default mode network and the ventral attention network, which is all about sort of responding to surprising things in the environment, surprising or salient things. We can break this down and look at changes across all of cortex. Here I'm showing all of the brain regions that show many changes, positive changes with age. So areas that show increased connections with age. And here I'm showing brain regions that show increases in negative or anti-correlations with age. And you can see that these regions are sort of all over cortex. It does look like there's pronounced remodeling in medial prefrontal cortex. We can also think about how cortex is segregated. So as I showed you before, in adults, we can see that you can break cortex out into 7 fairly stable different networks. In children, we use data from the ABCD data set. So we have, I think, several hundred kids, and we can ask, what did their networks look like? And this is what we see instead. And so I just showed you that we saw remodeling of medial prefrontal cortex over childhood. What we see is still at age 10, this area that is default mode in adults is limbic in children. It's sort of consistent with this idea that children's brains have more space for emotional processing. In contrast, visual and somatomotor regions seem to be sort of more stable, even by age 10. Another way to look at this is to think about how confident our algorithms are in this network assignment. So here I'm showing areas in pink where confidence is higher in adults. These computational methods more reliably classify these regions into the same network time after time. And there are very few, if any, regions where confidence is higher in children. So the story of development seems to be about increased functional specialization, increased functional stability. Right? The point or the goal or the sort of involvement of different cortical areas becomes stabilized. We think it's likely then that the last step is going to be gluing these networks into place through myelination. And the way we've been looking at myelination is a ratio of signal in T1 weighted images compared to T2 weighted images. This is called a myelin map. It is not really. It's really just how much stuff there is in cortex. I'm going to show you very new data looking at the development of this ratio in macaques and humans. We have data in juvenile macaques, and we have data that's now public from childhood all the way until age 36 in adulthood. What we can see in the macaques is that these brains turn from green to red pretty quickly. So by about age 3, we see adult-like levels of myelination. The process in humans is much, much slower. But you can see by early adulthood that sensory and motor areas are more highly myelinated than association cortex. I just want to plot those trajectories for you now. Here we have macaque development, and here we have human development. And I'm just showing you 3 curves. So sensory motor areas in yellow, mid-rank areas in orange, and association areas in purple. Across species, association areas are more lightly myelinated. We think probably more plastic. Sensory areas are more heavily myelinated. If we focus on adults, we see what we expect, which is some sort of plateau-ish around age 25. I expected this to be starker than it is. You can actually still detect a positive slope until age 40. Right, so these data are consistent with this idea that there's sort of a lot of gluing into place until adulthood, but maybe the experiences we have even in adulthood that build our expertise continue to lead to myelination. And this whole process of myelination in humans takes much, much longer than in any other species. So what I've shown you so far is that we see that inhibition plateaus in middle childhood. We see that functional specialization and association cortex continues past age 10 at least, and that the process of myelination, of gluing these networks into place, continues into adulthood.
Dr. McKay, we're going to switch interpreters now.
Thank you. Okay. So now we can talk about how experiences might impact the rate of maturation. And so far, we haven't measured experiences directly. Instead, we tend to rely on a proxy measure called socioeconomic status, or SES, that includes income, education, and occupation. What we know is that low SES creates risk for experiences like housing and food instability, exposure to violence, or low access to educational resources. It also creates risk for exposure to environmental toxins. And so it's sort of this cluster or constellation of experiences, but we're looking at risk rather than certainty. Say this field has also looked at a construct where we count up particular experiences called adverse childhood experiences. And this approach comes from public health, where people have noticed that the more of these experiences children have had, the more at risk they are of pretty much any health condition you can think of. Asthma, cancer, obesity, mental illness. And this centrality of ACEs in public health has led us to look at these in human development as well. And finally, we want to get a little closer ultimately to what kids are actually experiencing, okay, by looking at individual differences in social support and caregiving experiences. When we first started looking at these ideas, we focused on cortical thickness, or the distance between the outside of the brain and the gray-white boundary, as a way of looking at brain maturity. And we found that in adolescents, adolescents with lower incomes had more adult cortex pretty much everywhere in the brain. Their brains looked more mature. More recently, we found that in children, these differences by income are much more limited. So we think that they emerge over the course of childhood. But if we really want to understand how experiences relate to the pace of maturation, it might make sense to look outside of the brain, because we'll never be able to scan everyone. And this observation of accelerated maturation in low-income contexts is matched by observations of the body. So individuals from lower income backgrounds tend to go through puberty earlier. They tend to show faster epigenetic aging, so faster shortening of telomeres, the end caps on chromosomes, and faster changes in methylation. The problem with using these approaches for understanding individual differences in maturation is that puberty happens too late, and epigenetic measures are not reliable enough to use in individuals. And so these limitations led us to look in a different direction, which is teeth. The idea to look at teeth came from evolutionary biology. Molar eruption has been used for a long time to track the length of childhood across species. Here I'm plotting lemurs, macaques, gibbons, chimpanzees, and humans. And in purple, I'm showing the infantile period, or the time to our first permanent teeth. For humans, okay, we grow up the most slowly. This is about age 6. In comparison, we can look at the juvenile period, the time to our last permanent teeth. which is about age 20. And so I think these teeth maturation milestones sort of map to our understanding of changes in development. But what we wanted to know was whether there's individual differences in when people get their first molars. And so we decided to use data that we already had access to, our MRIs. So I'm going to show you what teeth look like at MRI. Here is a 4.5-year-old. You can see their molar is still in a bright sac. Here is an almost 6-year-old and another almost 6-year-old that differ in how mature their molars are. And here is a 7-year-old. Their molar is fully mature and in occlusion, meeting up with the top tooth. Okay. So now we want to check that this MRI measure matches clinical norms. So here we can see with age on the x-axis and molar maturity on the y-axis that older kids have more mature teeth, as we expected. And then we can look at how far each individual falls from the trend line. Right? Are they old or young for their age, and ask about individual differences. So as is expected clinically, girls have older looking teeth than boys. But the real question was whether this measure related to income and adverse experiences. So first I'll show you income. So individuals from higher income backgrounds have younger looking teeth. And individuals who've experienced more adverse childhood experiences have older looking teeth. And we were able to replicate this income association in a large epidemiological database. Okay, so now we can ask some really interesting questions. We can ask about how maturation of the body relates to maturation of the brain. These molar measures will be available in all sorts of public data. Because we don't need MRI data, we can look at dental record data to ask how disparities in maturation vary over space and time. Are there places with less severe disparities? What is different about those places? Were there times over history when the disparities were bigger or smaller? And we can ask about how interventions might influence the pace of maturation. We suspect that molar eruption might mark something like accelerated aging. If that's true, then we might be able to tell whether interventions are going to slow down aging processes, even in childhood, without waiting decades. So this line of work shows us that low family income is associated with faster maturation of the brain and the body. So next, let's switch gears to talk about motivation. And here I'm going to talk about motivational behaviors rather than the brain. And I want to capture individual differences in sort of parenting experiences rather than looking at socioeconomic status. So I want to show you the first experiment looking at variability and persistence in the lab. We have parents and children come in. We picked a task we know is too hard for them, and we just tell parents, let's see how many of these problems your child can do. Then we can code their parenting behavior. And I'll let you read the things that we coded. And we asked how each of these parenting behaviors related to child persistence as measured by parent report. And this is what we found. Of all of the behaviors that we coded, only taking over was correlated with children's persistence. So parents who reach in and take over for their kid more rate their children as less persistent. And this correlation could arise in a number of ways. It could be that impatient parents have impatient kids. It could be that parents who know their kids are impatient or not persistent will help more. But we thought what could be happening is that when parents take over more, their kids are less persistent. So we wanted to do an experiment. In this experiment, children were randomly assigned to one of three conditions. In the taking over condition, the researcher says, this is hard. Let me just do it for you. And this happens 2 times. In the teaching condition, the researcher patiently teaches the kid, says something like, what color is this block? And then there's a baseline condition where the child doesn't interact with the researcher at all. And then we ask, how hard or how long will a child try on a novel task? This task is a glued shut puzzle box. There's something inside it, so it sounds like it's possible to open it, but it's not. And we tell kids that whenever they want to, they can switch to this very fun looking water jet toy. I'm now going to show you a video of 2 participants, one that's in the taking over condition and one that's in the teaching condition. And then you'll see how long the child persists, just as 2 examples.
All right, so why don't you try to make the blocks look like this picture?
I'll pause so the interpreter can translate. Thank you. Oh.
All right, so why don't you try to make the blocks look like this picture? This is pretty hard, huh? Why don't I just do it? Does that look like the picture? All right, great. Let's try to make the blocks look like this picture. This one's hard, huh?
I need a whole white one, so this must be it.
Why don't I just do it for you? Does that look like the picture?
Yeah.
That's great. All right. So now it's time for you to play with a different toy. This is your toy. Sounds like there's something inside of there, doesn't it? I wonder if you can open it up. Why don't you play with it and see if you can open it up, and if at any point you feel like you can't do it, you can go and play with this other toy and I'll come help you, okay?
There's something inside.
Let's see if we can put these together and make them look like the picture. So why don't we start, with this top row. That's right. And then let's come down here. So what do we need for like that block right there? You see a red and white one we could put there?
That one?
Let's put that one in there. Awesome. And then we just have one left. And does that look like the picture? Look, you did it. All right. Maybe one more. All right. So same same thing. Why don't we start up here? So do you have a red one you could put in the puzzle? Okay, let's put that one right there. Okay, what about one that goes next to it? Do you have a red and white one you could put in the puzzle? We're actually gonna put that over here. And then what we're gonna do with this one is we're gonna turn it a little bit so that it looks like the one in the picture. So can you turn that block? And one more time. That's right. Okay, so now we're gonna look at, could we make one of our blocks red and white so that it could go right there? I'm gonna put it right there. And we're gonna do the same thing. We're just gonna turn it a little bit so that it looks like the picture. So go ahead and turn it. That's right. Okay. And then we just have one more to go, and we just need a white one to go right there. And does that look like the picture? Look, you did it. All right. So now it's time for you to play with a different toy. This is your toy. Sounds like something's inside of there. Wonder if you can open it up. Why don't you play with it and see if you can open it up? If at any point you feel like you can't do it, you can play with this other toy and I'll come help you. Okay?
Okay, so I just showed you two examples, a child in the taking over condition and a child in the teaching condition. Let's look at the results on average. When children don't interact with a researcher at all, they will try on that glued shut puzzle box for about 2 minutes. In the teaching condition, they also will try about 2 minutes. But if they're in the taking over condition, they give up after just 1 minute. So I will share that when I was working with Julia Leonard, who's now faculty at Yale, on this graph, I offered to make it for her because she was struggling. And she just looked at me and said, "Have you learned nothing at all?" It is so easy to take over for people, even when you have the best intentions. And it's hard to watch people struggle. But now I pause and reflect and think, what are people learning when I take over for them? My children and people in my lab. It might make them infer that I will help them always, or that I don't think they can do it themselves. Okay, so this line of work is on sort of individual differences across children and persistence. But we also know that there's variability in persistence within child. So I'm going to show you data on a child brushing her teeth on a good day. So you'll see
Good job.
that she worked very hard.
Get the top, Stu. Other side, honey. Good. A little longer.
Okay, and then the very next night, this is what her persistence looked like.
Feeling a little sleepy. A little longer, honey.
No, you.
Can you try just the back? And the other
Yeah.
side? In the front?
Okay, so if a child came into the lab on this day, we would have said this is a very persistent child. Not so much on the next day. So where is this variability coming from? So we ran a study where we asked daily questions about child sleep, child mood, and parent stress. And we had them send in videos of nightly toothbrushing. We have this data for 2 weeks on 86 3-year-olds. We chose toothbrushing as a way to measure persistence because it's something kids have to do at the end of the day when they're tired. It's something where you just have to persist. It's not difficult, but it's also not interesting or exciting. It's a little bit of a boring task, but it's one that parents really want their kids to do. So let's look at the data. We see that variability in parenting relates to variability in child persistence. So on nights when parents used more praise, their children brushed longer. This is not the same as saying that parents who use more praise have more persistent children. It's saying that within a child, within a family, when parents use more praise, their kids will brush longer. And we looked at the timing of that praise, and it's not just that there's praise at the end for a job well done, but there's this sort of reciprocal relationship between child behavior and this positive feedback. If you count instead how much direct instruction parents are giving, saying things like brush your teeth, you get the opposite relationship. So on nights when parents use more direct instruction, their kids have worse behavior. When we look at some other features, we see less reliable relationships. In general, kids will brush longer when children are in a better mood. But not everyone. There's no consistency about the relationship between child sleep and persistence, which I found shocking as a person who's very sensitive to sleep loss, and my children are very sensitive to sleep loss. That's not true for everyone. And similar variability and sensitivity to parent stress. Okay, so here we see that caregiver behaviors are linked to and can alter motivational behavior. So I want to close by talking about some thoughts about how we might be able to enhance brain plasticity. So first, we can think about how we can harness motivational systems, boost that dopamine. Okay? We can take over less. We can praise more. And I think critically, we can identify the factors that shape motivation in each child. And I think we can broaden this out. We don't have the evidence for this yet, but I think even for adults, we can think about what matters for us. Returning to the neuroscience idea, I think it's useful to map out these axes so that we can time interventions when plasticity is high but waning. You wouldn't want to intervene here because anything that you do could be quickly undone. If you intervene too late, it might be hard to change the system. But if you intervene here, the changes that you make might get glued into place. But of course, we think some of these disparities start really young. So we can also think about different approaches to supporting families of young children. So right now we have an intervention going on called the Brains and Early Experiences Project, where we have 200 kids that were randomly assigned to either receive a program called Parent Child Plus, where they get almost a year of 2 visits a week with new books and new toys and parent coaching and support at every visit, as compared to a program that's around family nutrition, where families get money for groceries. Right? And we can then ask whether the set of experiences from Parent Child Plus changes things like inhibition, functional specialization, or myelination. And do these positive early experiences boost brain plasticity? We can also ask if this program slows down maturation of the body by looking at molar eruption, or changes parent-child interactions as measured with toothbrushing. So sort of all of this foundational work we've put into figuring out good outcome measures, we can now put to use in the context of this very hard to run, but very exciting intervention project. And the last approach that I want you to think about is who needs to know this information? Who should we share the science of brain plasticity with to change outcomes for young people? So last month, I worked with an organization called Spring Point Partners to bring the UCLA Center for the Developing Adolescent to Philadelphia to create trainings. I took this picture of a training for members of City Council around brain plasticity in young people. We trained the police, the DA's office. We trained people who employ young people. We're now working with the school district to talk about what it means to have a plastic brain. What are the costs, but what are the opportunities? How can we make things better for our young people? And with that, I'll say thank you, and I look forward to your questions.
Thank you so much for your presentation. So we will now invite anybody who has questions, if they would like to type those questions out in the Q&A feature at the bottom of the screen. They may do so now. And if a person prefers to ask their question in ASL, please send a chat message to me and we will promote you to panelist so that you can ask your question in sign language. Well, people are still thinking about and typing out their questions. I do have a few. Was curious about the molar eruption measure that you described during your talk. Is that measure showing a shared mechanism of general development, or is that a proxy for SES? Because I'm thinking maybe lower SES individuals will brush their teeth more often, and maybe their baby teeth will start falling out at a younger age, which will then encourage their adult teeth to grow in earlier. So just wondering what your thought is about that.
It's a really interesting question. What I will say is that we're looking at molars when they're beneath the gum line still. So I don't think it is about any kind of brushing habits. We don't know that for sure, but generally what happens is you see a change in molars before you start to see teeth falling out. That's sort of the order of events. We have looked at sort of what else it could be. So in the epidemiological data, we asked whether molar eruption was correlated with something like lead exposure or nutrition. That's another thing people think about a lot. We don't see those correlations. We do see, if we look at the 12-year molars instead, we see a correlation with onset of menses or pubertal development. And we see even in the young kids correlations with adrenarche hormones. So it does look like there's something about the pace of maturation more broadly. I will also say that we see, I sort of ran out of time for this, but we see surprisingly strong correlations between molar eruption status and cognitive development in a way that you might not expect, even controlling for SES and controlling for adverse childhood experiences, controlling for race and sort of experiences of racism. And so what I think we're picking up is something like how much of that stress kids have internalized. And so kids who are more impacted by those experiences in terms of their molar eruption might also be more impacted in terms of their cognitive and brain development. But that's an open question.
Thank you. All right, I do see a question on the Q&A. Dr. McKay, did you want to read that for yourself?
Sure, I'll read it. So in the video I shared, did I account for racial ethnic power dynamics between the facilitator and the child and family? What's really important about that experiment is it's randomized in terms of who is assigned to which condition. Right? So we did sort of check to make sure that the two conditions were matched at the end in terms of racial identity of the kids. But in this particular case, that can't drive the effect of the experiment. When we look at correlations between taking over and children's persistence, we did check to make sure that we could control for family income, race, ethnicity, child age, and gender, and still see that association. But these dynamics I worry about more in correlational studies than I do in random assignment.
Just a follow-up question for you related to that particular study and the overall results about children's persistence. How much do you think culture and where the actual study happened had an effect? Because I think there's more and more evidence that shows where our experiment takes place will impact people from different cultures, different ways, have a way of managing parenting and how they interact with their children. And just thinking about, will those have an actual impact while interpreting results?
I think that's really important. And it's a reason that we've moved away from doing parent-child observations in lab, because I think it's absolutely right in a way that's going to ever be hard to control for. So we can control for things like race or education or income and think that we're getting rid of some of this cultural variation, but we're not, right? It's still unclear how parents feel about being in a neuroscience lab and how that changes their assumptions about what the task is for and what we're looking for. So I think it's really complicated. And another reason that when we found the association between taking over and children's persistence, that we sort of drew inspiration from that for the experiment. What I am excited about now is the toothbrushing study, so we can sort of understand parents' behavior in a more natural context. So there's still some question about what parents think we're looking for, but we sort of see over time that parents sort of talk less over 2 weeks, right? So at first they're really trying their best and sort of guessing more about what we're trying to look for. 2 weeks in, it just becomes very routine, and it starts to really feel like a window into homes that is less, the parents are thinking less about who we are and what we're seeing.
I want to go back to the brushing study. So how do you handle confidentiality and privacy with getting all of these videos in settings where people, are in their private bathroom, maybe dressed, maybe not dressed, showing you their private life? How do you handle that?
Yeah, it's a good question. We do a lot of training up front around how important it is for children to be wearing clothing. We learned that because a colleague of mine was sort of our first pilot participant, and her kids were very naked. Right? She just was not paying attention to putting clothes on her kid. And we're like, oh, okay, this is a thing that we really need to pay attention to. So they get a lot of training. We have sort of cartoon, Elmo is our character, of sort of like what a good video looks like, what a video is that we can't use. And then sort of over the 2 weeks, we give parents feedback. In the context of the intervention study, we have, because these families are very, very low income or sort of dealing with more instability, there's sort of more variability in who's with the kid, brushing the teeth, and sort of where the kid is and what's going on, time of day, all of this. And so we're sort of constantly working to make the paradigm more inclusive, so we can sort of better understand variability in kids' experiences. The videos that I showed you today are from my kid. So I have consent to use those videos. And we do have a set of videos where parents have given us the right to share those videos, but we're a lot more careful with those videos than we might be with de-identified MRI data or something like that.
Of course. I do see another question in the Q&A.
So this question is about how the Hawthorne effect is accounted for in the intervention study. I don't think we know yet who will benefit most from this study. So working with Parent Child Plus is a community organization, and the funding to deliver the intervention itself has restrictions on it placed by funders and by government sources around who is eligible. So families have to be low income to participate. And they have to be in high priority neighborhoods of Philadelphia. So they have to not earn a lot of money themselves, and they have to live in neighborhoods where there's high levels of disadvantage. So there is less range than you might have guessed in terms of the sort of incomes of families who are participating. So I don't know. It'll be really interesting to look at those individual differences to see who will benefit most from that kind of experience. Then the next question is around limitations of using persistence as a measure of performance. In some situations, it might not always be better to persist in an environment. It's a really important question. What I think about in terms of what is good persistence is what is effective persistence, right? And a lot of what kids need to learn is when to persist and when not to. So if we think back to the child who was in the taking over condition, it was very adaptive for him to give up, right? He just got information that tasks are going to be too hard for him or that somebody might help. And so the adaptive thing for him to do is to give up. So when I say, what does it take to boost motivational behavior, I'm not really interested in making sure kids try hard all the time, just that they are able to try and to sort of get the neuromodulatory boosts from dopamine when it makes sense to try. We have a parallel line of work on boosting curiosity, and it's the same kind of thing, right? You don't want curiosity all the time. It's not adaptive to ask questions all of the time or to seek more and more new information. You probably have had the experience of scrolling through Twitter or social media where you're getting information all the time, but maybe that's not the most adaptive thing to do in that context. So I think it's really about how do you tailor motivational behaviors to the context. So the next question is around, is there more research out there with deaf-related research about persistence? So Julia Leonard, who led the work in my lab as a postdoc, is now running her own lab at Yale, and she has a lot of sort of new lines of work on persistence. I don't know if she has any work right now that is deaf-related, but I think she'd be excited about your ideas. So if you reach out to me, I will help you get in touch with her.
I want to go back to one of the larger themes that you've touched on today during your presentation. It seems that there is more and more evidence showing how negative life events encourage maturation or early maturation in children. Now, if that happens, I'm sure that it has a purpose. And so can you talk a little bit about why that happens or why that mechanism would, or how that mechanism can be adaptive for children?
Yes, I started out this line of work thinking that it would be very adaptive. Childhood is a time of vulnerability. It's a time when you are reliant on many other people. And so it makes sense to me that evolutionarily, signals around early life stress would signal grow up fast. We don't have the luxury of a long childhood in this particular context. And in a lot of contexts, that would be great. You would sort of build adult level skills earlier in life and be able to contribute earlier. But we're asking kids to sort of be selected into careers based on high school calculus and geometry, right? So they're sort of running up against an education system that assumes high brain plasticity well into adolescence. And so I really think it's about a mismatch of how brains and bodies are adapting to experiences with sort of what our society demands. I will say that I am still suspicious that this accelerated maturation phenomenon is not all adaptive. So we know, for example, that the maturation of molars is not strongly correlated with lead exposure. In some groups, it is correlated with cadmium exposure. which comes from smoking and like secondhand smoke exposure, and some pollutants. Need to learn more about cadmium. This is very new data. So I think the pace of maturation can be shaped through adaptive processes. I think that there might be other processes that we didn't evolve to experience that are causing things more like aging. The other set of processes that I haven't ruled out are immune-related processes. So individuals from low resource backgrounds are more likely to ride crowded public transportation, for example, or work in places where they have lots of germ exposure. So I still think it's possible that people who are exposed to more pathogens or get sick more often go through earlier aging processes, and I haven't ruled that out yet.
Thank you. Yeah, I didn't think about how pathogen exposure would impact maturation.
I'm not sure it does.
All
It's
right,
something I want to look into.
I guess we'll see, right?
Yeah.
I mean, if you think of the range of possibilities, everything that impacts our bodies. And often chronic illness can become very stressful on the body. It has other stress-related consequences, so why not? I don't see any other questions in our Q&A. So I think your presentation was very clear, very interesting. Look forward to seeing more of your work come out of your lab. And thank you again for giving this presentation here at Gallaudet. I think we can close out here, and if anybody would like, they can get in touch with me, and I will try to make sure that I pass on any of your research articles that would be helpful for them in their research.
Thank you so much.
All right,
Please
thank you,
let me know if you
everyone.
have other questions. Thank you, everyone.
All right, and thank you to our interpreters and our captioner as well.
Yes, thank you.
Bye, everyone.